Thank you very much for the introduction, and thanks for inviting me. It's really a pleasure and an honor to be here and to speak. And you see, this is not my actual title. This was my initial title. I wanted to talk a little bit from the clinician scientist perspective on what we are doing with respect to translational medicine in Gießen. However, so this is, no, here you go. This happened. And I was asked to change the topic a little towards more antiviral or research in antivirals. And this is why I changed the title to From Infection Research to New Antiviral Drugs. And the first part of my presentation will be on what we are doing in the lab with respect to new antivirals against coronaviruses. This is still preclinical and has not reached the patient, but we hope that this might happen. But still, I will go and I will talk a little bit about my perspective or why we do need clinician scientists in that process at the very end of my presentation. So let me start with highlighting the importance of this research topic on virus-induced pneumonia or virus-induced lung injury. It is well known, and this has been published by the ECDC in 2018, that lung or respiratory virus infections pose a major threat to human health and have a high disease burden. And this is shown here. This is depicting the annual cases and the annual deaths of the, let's say, 25 most important infectious diseases in European countries. And you see that influenza ranks highest with respect to cases and also with respect to deaths. And the bubble diameter depicts the number of disability-adjusted life years per 100,000 in the population. However, it's not only about viral infections, but we know, and the clinicians among you know, that respiratory viral infections are frequently followed by bacterial infections. So they pave the way for severe and invasive bacterial infections, such as IPD, invasive pneumococcal disease, and in kids, Haemophilus influenza. So then there was a paper in 2015 in the New England Journal by Seema Jain's group and the EPIC study team, and that was quite surprising because when this study team actually analyzed the pathogens that could be detected in patients that were admitted to hospital with severe pneumonia, among the 7 most frequently detected pathogens, there were 6 viral pathogens, and that was really surprising and a game changer in the field. And you see that the most frequently detected pathogen is rhinovirus, followed by influenza, and then followed by Streptococcus pneumoniae. But then there's also human metapneumovirus, RSV, parainfluenza, and coronavirus. So it was evident from that time on that respiratory viruses account for a large number of severe pneumonia cases worldwide. And we also know that there is increased susceptibility of patients with chronic lung diseases that are typically triggered and often exacerbated by viral infections. And those are mentioned here, and you know that COPD and asthma have a high prevalence in the European and US population and also in Asia. Now, I do not have to highlight that emerging respiratory viruses are also a problem in these days. And I mentioned the new coronavirus here and also SARS and MERS coronavirus and highly pathogenic avian influenza viruses such as H5N1 or H7N9 that some of them come with high pathogenicity and also with pandemic potential. And I've highlighted this here again because this is the major topic in these days. In striking contrast, there is high medical need for research in that field because there are only few antivirals available with limited efficacy and only for influenza. There is no causal treatment of the most devastating course of pneumonia, which is the acute respiratory distress syndrome or ARDS. This is pneumonia-derived lung failure or followed often by multi-organ failure. Then it's depicted here. This is a patient with H1N1 influenza. And then the influenza vaccine has to be adapted each year and comes with limited protection, and there's no vaccine against all the other respiratory viruses available. So we need research in that field. And this is why in the last 15 years we focused on asking or answering the question why distal lung virus infection often results in severe damage of the alveolar epithelium. And we identified several pathways and molecular mechanisms by which alveolar epithelial injury in the distal respiratory tract is triggered, and this is often by exaggerated and unbalanced host responses. And you can see one important factor here. This is a pro-apoptotic ligand, which is released from macrophages that are recruited to the lungs upon severe influenza infection. It's an interferon-dependent gene which is directly killing or rendering apoptotic alveolar epithelial cells, and not only cells that are infected by the virus, but also cells that are uninfected. So TNF-related apoptosis-inducing ligand is crucial in triggering severe lung injury and barrier failure, and we also identified some other candidates that we would like to target as for with therapeutics in future. Now, in the last years, we also focused on mechanisms of inflammation resolution and epithelial repair, and we identified just recently that so-called bronchoalveolar stem cells, or BASCs, that are precursors and endogenous stem cells of the lung, precursors for bronchial and alveolar epithelial cells, are crucial in repairing the lung following a viral infection. But we also found that if they are infected by influenza virus, or if their niche gets signals from an influenza virus infection, this might also result in aberrant repair, and this also highlights that gives us the possibility to actually target such processes for better repair after severe viral infection. Now, I would like to show you some of our strategic aims. And first aim is to gain better understanding of the molecular interactions at the virus-host interface to design more effective antivirals, at best with broad antiviral capacity, and I will show some unpublished data on that with respect to coronaviruses. And then we try to elucidate how viral infection drives both structural and functional damage to different cellular compartments of the distal lung, and how the infection impacts mechanisms of injury resolution and lung regeneration, both in immunocompetent hosts and in patients with pre-existing chronic lung disease. And finally, of course, and that's why we are here, to integrate and translate these findings into novel treatment strategies, including host-based interventions, and my second example will highlight such a strategy towards first-in-human trials. And now back to the current situation. The new coronavirus, which has now a new name, it's called SARS coronavirus 2, because there is some identity to the previous SARS coronavirus 1. Just very briefly, you all know that data, this is data of January 23rd this year, with only 500 lab-confirmed cases, a few deaths. However, in February 11th, mid of this week, we had over 40,000 confirmed cases and over 1,000 deaths, people, all of them except one in mainland China. The disease caused by the new coronavirus is called COVID-19. It's just simply coronavirus disease 19. And some facts on that. It was in early December last year when in Wuhan there was an accumulation of some pneumonia cases of unknown origin, and some weeks later, end of December, experts from the Chinese CDC arrived in Wuhan and informed the WHO about that process. And they said that there was probably a primary infection at a local animal market, but they were quite sure that there was no human-to-human transmission in these days. Then a week later, there was the official announcement that this was caused by a novel coronavirus of the family of beta coronaviridae, and that was approved by the WHO 2 days later. And just 7 or 6 days later, the genome sequence was available and published, and this was very helpful because there could be, a diagnostic test was rapidly available then, first one published on January 16th. And it is, as already mentioned, quite evident that this virus is similar to the previous SARS virus. there is around 90% sequence identity to a bad SARS-like coronavirus that has been the precursor of the SARS virus, and 80% identity to the SARS coronavirus 1, and they share the same receptor for cell entry, which is angiotensin angiotensin converting enzyme 2, ACE2. And now, of course, it's evident, and we all know that human-to-human transmission occurs, and we can detect virus particles in mucous membranes, blood, and stool of infected patients. Very likely there is transmission via droplets, and other routes have been discussed but have not yet been confirmed. And the major issue, everyone is talking about that in these days, is that incubation period is quite long, 2 to 14 days, and it seems that a lot of patients are oligo or asymptomatic during that period but can still spread the virus. And epidemiology-wise or containment-wise, this is of course a major issue. Now, as said, the clinics is asymptomatic to severe pneumonia. Frequently patients have fever and cough, but some are oligosymptomatic as shown here, and the fatality rate is given with 2%. However, it is likely that it's a little lower because many infected patients are not discovered or not diagnosed because they are A or oligosymptomatic. Now, question, of course, are there any specific medicines to prevent the new coronavirus? And the answer is no, there is only supportive care available to date. However, we try to solve this. And then, of course, we wanted to go into a preclinical animal model, and that was a little bit tough because mice do not express the receptor, which is relevant to take up MERS coronavirus into the cells. So we teamed up with some researchers from the German Center for Infection Research, and also with a group here from Berlin, Achim Gruber, who is at the FU. What we did was we established adenoviral transduction of the human DPP4, this is the receptor, with an M-cherry construct by intratracheal application, followed by infection with the MERS coronavirus, and that worked quite well. So you see that over 90% of the alveolar epithelial cells, or the lung epithelial cells in mice, in fact express the DPP4 receptor, and this is also shown here. And then you could easily infect these mice, and they express the MERS coronavirus spike protein here. Now, having this model established, we also characterized the pneumonia that happened, and I think this is something very relevant because we really need to know whether the model really shows what is happening in humans, right? So it should be similar, and that was what we found. There's necrotizing bronchointerstitial pneumonia, there's alveolar edema, and hemorrhage occurring. Within the German Lung Center, we have the possibility to collaborate with the Fraunhofer ITEM Institute for Toxicology and Experimental Medicine at the DZL partner site Brest in Hanover. And we are currently starting to establish formulations for aerosolized deposition and compound modifications, inhalation toxicology, inhalation devices for CSA and alisporivir in mice towards first-in-man trials. Now, this is the first part of the story, and as said, I present this because of the actual outbreak situation. This is not yet in the patient, but I would like to highlight that there is other things going on at the University of Gießen-Marburg Lung Center. And first of all, maybe just to show you, we have a successful translational pipeline for lung therapeutics established at our center. With 5 medications that finally made it from discovery at the bench, from in vitro trials, through proof of concept, through preclinical development in animals, phase 2, phase 3, towards worldwide approval. And another one is on the way. And then there are some phase 2 trials going on, and I would like to point out this one. This is inhaled GM-CSF for pneumonia-induced ARDS. Now, it's not only viral pathogens that cause severe pneumonia, of course, as I mentioned at the beginning, but there is also, there is a large unmet medical need for pneumonia per se, but may it bacterial or viral pneumonia. And this is a slide that I took from Norbert Sutop, actually, because I like it a lot, and this shows that the mortality of severe pneumonia has been unchanged within the last 70 years since the introduction of antibiotics, and it's estimated, or we estimate it's going to stay like this, or it might even rise again because of new emerging viruses or because of antimicrobial resistance, as the WHO stated a few years ago. Now again, an unmet medical need for adjunctive therapies beyond antibiotics. And I show this slide because the whole story started in 2006 with my first ever PhD student that I had, Lydia Shakarova. She started work in my lab, and she found that GM-CSF, which you all know, it's a very well-known growth factor for myeloid cell differentiation, activation, and survival. And it's in clinical use for patients with severe neutropenia, particularly in kids. That GM-CSF is also polarizing macrophages towards an M1 phenotype in vivo and drives host defense, activation of macrophages, but it also, and that was her new finding, it drives epithelial or epithelial stem cell proliferation and differentiation in the distal lung. And that was published in 2009 in the Blue Journal. Now, what she found is she used a model of gram-negative pneumonia with Klebsiella pneumonia that was applied intratracheally. And she used wild-type mice, GM-CSF deficient mice, and those mice here are SPCGM, and those are GM-CSF deficient, but have constitutive alveolar overexpression of GM-CSF in the distal lung, with high levels of GM-CSF in the lining fluid. And what you see is that after insult, may it be LPS or Klebsiella, in the GM-CSF deficient mice, there is no proliferation of the local stem cell pool in the distal lung, which is composed of type 2 alveolar epithelial cells. So there is no regenerative response. And if you look at barrier function, alveolar albumin leakage, in the GM-CSF deficient mice, this is sustained. So the mice cannot repair their barrier and get back to normal barrier function. We also analyzed this in an influenza model. We wanted to know whether this holds true for viral infection also. And we used the low dose of infection. This is an H1N1 virus PR rate where all wild-type mice survive. They get sick, but all of them survive. And the GM-CSF deficient mice all die from the infection. So there is a clear phenotype. And then we went higher with the infection dose with 2,000 PFUs where all wild-type mice die of the infection, but the mice that overexpress GM-CSF in the distal lungs, in the lining fluid, they survived to around 80%. And then we wanted to know whether it was the lung parenchymal, the epithelial GM-CSF, or the myeloid GM-CSF driving that. And we did bone marrow chimerics, and you see that you in fact need alveolar epithelial GM-CSF to survive a bacterial or a viral infection. And that gave us two pieces of important information. First of all, no matter if you have bacterial or viral infection, you need GM-CSF for host defense. And second, you need high levels of GM-CSF in the distal lung and the lining fluid to survive infection and to repair. So we thought we should give GM-CSF intratracheally and not systemically, so into the distal lung to deposit it locally. And this is what we did. And you see, this is for a viral infection model. We can activate dendritic cells for generation of T cells with better antigen presentation. We reduce viral load. And particularly, this is of importance, we reduce alveolar epithelial cell injury. This sustains alveolar barrier function, and this is the blood gases, so there is no or less respiratory failure if you have GM-CSF in your lungs. So this is the summary of the let's say, the preclinical findings. More or less, we know that GM-CSF drives host defense functions of macrophages, both in gram-negative and gram-positive pneumonia. We know that it drives local expansion of CD103 dendritic cells for generation of antigen-specific T cells and viral clearance. And also, GM-CSF drives proliferation of different epithelial progenitors in the distal lung for epithelial repair. And we thought these manifold beneficial effects should be used to establish new therapies by GM-CSF. Now, during these years, we actually had some patients on our intensive care unit that were on the ventilator with severe pneumonia or pneumonia-associated ARDS and had their antibiotics, but did not really recover or did not get better after a while because they either could not get rid of the bug or they could not properly repair their lungs. And within a compassionate use, we had them inhale GM-CSF twice within 48 hours. And as we had an established registry of pneumonia or severe pneumonia and ARDS patients, we were fortunate because we had this infrastructure around, which is provided by the German Lung Center, and this is the Gießen UGMLC Biobank, which is part of the large biobank of the German Lung Center. This is just indicating the samples that we have. This is last year, February, so we have a lot of pneumonia ARDS samples in this DZL wide biobank. There's a lot of CAPNet samples in there, of course. And this is connected to a data warehouse where we can retract the phenotypic data of the respective patients from these samples. So this actually gave us the possibility to do a bedside to bench approach because we had samples of these patients in the biobank. That was really instrumental because what we found was that indeed pulmonary macrophages in these patients get activated. They upregulate, for example, CD80 or HLA-DR, which are classical activation markers for host defense. We also looked at other markers, but also we saw that, and this was quite a low number of patients, so it seems maybe we were lucky, but this is a cohort of non-treated patients that we used for comparing that, but oxygenation after GM-CSF treatment was better within 10 days, and we also had a reduction in morbidity scores after GM-CSF treatment. So we published that, yeah, this was the state where we were, and then sometimes you end up in Death Valley #1 in the translation because now you really would need to go for a clinical trial, but who is going to fund that and how to implement that? But as mentioned, we published that, and we were lucky that there was a company that read our paper, and this was a small Danish company, Serendex, that was just on the way to prepare an inhalation version of GM-CSF for ARDS. And we started to team up with Serendex, and they had the preclinical testing for inhaled GM-CSF in monkeys already done and just started a phase 1 in healthy volunteers. So we were really happy with that. We started the GI Hope trial, a randomized double-blind placebo-controlled multicenter phase 2 trial. This is the protocol. Patients get BAL to phenotype as primary endpoint the macrophages, and I will get back to that, why we do that a little bit later. This is the inclusion criteria, and of course we have secondary endpoints that are more clinical and focus on oxygenation, barrier function, and so on. So this was posted on the Serendex website in these days. Great beginnings with Gießen, but unfortunately it had a rapid ending when Serendex was dependent on venture capital, but they didn't make it, right? So they were bought by a US company called Savara, and Savara continued the GM-CSF program for pulmonary alveolar proteinosis. This is another indication for inhaled GM-CSF, but they withdraw after a while the support for the ARDS program because they had a stock market launch, and they said investors would never accept an ARDS program with us. So we were lucky that the German Center for Lung Research within its clinical trials program started up to fund us again. So we are now again running as an investigator-initiated trial within 3 DZL sites and 3 non-DZL sites. We are recruiting the final patients now, and we were lucky to get an additional funding for a bedside-to-bench research program where we do single-cell RNA sequencing and profiling of the macrophages of patients before and after GM-CSF or with high versus low dose of GM-CSF or placebo. And we do pseudotime analysis to map trajectories of macrophage activation and macrophage differentiation in these patients. So at this point, I would like to summarize with my experience, what have been the key elements and the key infrastructures that were relevant to enforce translation of this story. And I would like to start with this, this is a citation of this publication from Duda et al., and that was after the Translate meeting in 2014. And he said, or the authors say, that basic biomedical research is really important to drive the discovery engine. And I cannot highlight this enough, because we were lucky to have all the relevant preclinical and lung infection disease models in place, from like organotypic cell cultures and organoids to rodent models towards biobanking and first in humans. So I think this is crucial, and we are happy to have a substantial number of funding within the lung research field. So then, of course, we were somewhat lucky, because this was a repurposing approach in both cases with the CSA and also like with the GM-CSF. So we had the phase 1 rapidly accomplished for new application route, which is the inhalation. And within the DZL, we have the chance to collaborate with institutions that help with modifications or lead or compound modifications and also establishment of first in man. Now then, the famous mindset for translation. That was for sure present, or is present, at our center with the translational pipeline, and I've shown that slide before, and a local culture of building private-public partnerships that is successfully or very successfully established in the PH field, as indicated here. But these 5 medications here are, of course, only the tip of the iceberg, and many others failed, right? And we also had ARDS trials that were in phase 3, which also failed. So there needs to be some kind of failure acceptance in that. So you still need to drive it, but you need to know that it might fail, and you need to talk to your PIs or to your people and tell them, hey, it's no problem if you fail. This happens. This is something which is normal on the way from or to translation or during translation. We have a nice structure which is called Transmit. It's a small company which is affiliated to the Justus Liebig University, and they help a lot in patenting and technology transfer issues in the value creation chain. And this was not so relevant for GM-CSF, but it's now relevant for a new substance that we want to drive also during or through that pipeline, which is a repair factor that is expressed from macrophages, and we are just, they were very helpful in patenting that. And then, again, this I mentioned initially, you need clinician scientists to drive that. And we have programs in place funded by the DFG, but also within the Excellence Cluster and the German Lung Center. And I also mentioned that the biobanking and data warehouse structure of the German Lung Center was instrumental for the bedside-to-bench approach. And this allowed us and will allow us in future for precision phenotyping and better molecular characterization, and therefore also for better protocol design of clinical trials and for better readout definition. And now let me come back to the problem why Savara did not want to fund our ARDS trial anymore. Because a lot of ARDS trials have failed. And I think the reason for that is that it's more or less a collection of different heterogeneous syndromes than a precise disease definition. And we lack definition of subphenotypes and a much better characterization, and we can only do that if we have such structures in place. And the CEO of Savara, he called me and said, you know what my investors say? They say ARDS is the graveyard of pharmaceutical industry, so get rid of your ARDS program. But we hope that this will be not the case anymore in future, of course. And then, of course, the DZL clinical trials infrastructure with funding rapidly available after withdrawal of support. So we need cross-funding still because this is an expensive trial. But still, we were happy to be able to continue that as an IIT now, and of course structures that support us, which are associated to that, clinical trials infrastructures for study design, protocol submission, and also trial management. So there's a lot of DZL, as you see, so that was really instrumental. But I mentioned at the beginning that I would highlight some aspects of clinician scientists in that relation. So maybe there are some clinician scientists in the audience, or some of you are on the way to decide whether they want to be a clinician scientist. And yeah, you can just say it's always a high wire act between lab and clinics. It's not easy. You compete with scientists all over the world that have high-ranking publications and that do not have the clinics at the other side. On the other side, your clinical colleagues do not regard you as top-notch clinician because you have all that free time for research. So they still call it free time. I don't know why, but so it's really not so easy. And this is how you might feel. I think there is no English word for that. Eierlegende Wollmilchsau. But still, although you might feel like this from time to time, it is evident that we absolutely need clinician scientists, not only on the way to translation, not only in the preclinical development, but we also need it later in the translational process. Particularly here in translation to practice and translation to community, because we need a much more deeper understanding of these medications or therapies of increasing complexity, and we have more and more personalized approaches in the clinics, and we will have them in future. So we need a deep molecular understanding of disease, and therefore we actually need clinician scientists. So that is my final slide, and I would like to acknowledge all the collaboration partners here, the funding sources of that research, and thank you for your attention.
Yeah, thank you very much for the stimulating talk, and I'm sure there are questions from the audience. Yeah, please.
Yes, so we both sitting here together have been involved in the development of mavacamab, which is an anti-GM-CSF receptor antibody. Actually, we suffered the same fate as you did because the company finally pulled out of that program.
Right.
But we treated several hundred patients with rheumatoid arthritis with this compound, and it was remarkably safe. So we didn't see any infections. Maybe modeling showed us that not too much of the antibody reached the alveolar space, but still was quite safe.
Right.
We didn't see any infectious events.
Right.
So maybe there is a more, let's say, different situation in ARDS that we just have a special situation where we use that compound.
Yeah, right. And there is a disease called pulmonary alveolar proteinosis, and these people, yes, yeah, exactly. These people are not susceptible to infection, right? So they are not getting more infections than other people. I think the point is, and this is why we designed the trial in the way we did, that it's more or less the state where you still have maybe ongoing presence of some bacteria. You cannot really get rid of it. You come into a situation of kind of energy in the distal lung. So something we observe during sepsis when your immune system declines. So you cannot get rid of the bug. And at the other side, you cannot really repair. And you only need repair if you have injury before, right? So this is why we start GM-CSF inhalation only after a few days. So the patients get antibiotics. They usually they also start to get lower inflammatory markers, like CRP gets down, but they still need high oxygen, right? And one inclusion criterion is to have oxygen supplementation over 0.5, so 50% FiO2 still. So this is actually the indication we have exactly for that reason. I think it's relevant to have GM-CSF once the thing is already going on and it's not going better, right? That's what we think. So that's also what we see in mice.
Thank you. Okay. I wanted to follow up on the question from the other end. Is there a danger or risk of having too much GM-CSF? So in your SPC overexpressing mice, do you see any changes in alveolar morphology?
Yeah. They have a little more. They have more type 2 cells. Yeah. But they do not have ongoing or let's say overshooting presence of immune cells in the lung. And we have, of course, analyzed this in our patients, also in the phase 1 and in the monkey study. You do not see a high recruitment of inflammatory cells into the lung. So that was the main caveat, of course. So if you have an ongoing inflammation and you trigger myeloid cell development, maybe you have leak to the bone marrow if you inhale it still, right? So this was one of the main issues we wanted to look at, but it seems to be safe in that regard. And SPC-GM mice have, yes, they have higher numbers of type 2 oval epithelial cells, and GM-CSF mice have a little lower numbers, so they have some developmental defects. It's not very striking, so it seems that during lung development, mice can compensate with other, let's say, pathways and so on, but they have a little, some minor structural issues.
If the coronavirus binds rather than the H2 receptor, it would not just be a therapeutic option to block the receptor.
Yeah. Yes, this is an option, and I'm sure people will go for that. With DPP-4, you know, there is DPP-4 antagonists on the market for diabetes, but it's not working. It's not working. It's not working against MERS.
Did they already do molecular modeling where this maybe needs to be at the right position of the ACE?
Yes, exactly. Yeah, exactly. I mean, you need to design the proper, the proper target and mediator. But yeah, this is of course something that people do.
That's what's being from an HIV therapy to block.
Yeah, of course. Yeah.
So I'm meeting a molecular design chemist this afternoon, so I will ask him.
Right. Right. Yeah. But you need to know that. I mean, this would be a more preventive strategy, right? To block entry. In like when severe viral infection goes into the completely wrong direction, you have already reached your viral peak, right? And then people come and are like get severe pneumonia and have all these issues with respect to barrier function. So, and this is when the story is already running. So maybe I don't know whether entry will be more a kind of preventive strategy, but still, of course, this I think people will go for that. Yeah.
Well, I think I never understand. I think there's a lot of debate. So where do these patients, let's say, with the coronavirus die from? Is it really the viral load, or is it the immune system that ultimately kills them? Yeah, because,
we don't know.
I mean, nowadays with all the plethora of drugs, JAK1 inhibitors and so on,
right,
could tackle that end, but maybe that's too late, or what is the word?
Right. For the new coronavirus, we don't know still. But for SARS and also for MERS, it's exactly what you say. It's not so much the viral peak and the injury directly from the virus, but it's more or less the injury by the immune system. And this is definitely the case for influenza.
Which we could tackle actually, the modern compound, like JAK inhibitors, for instance.
Right, yeah.
It would be interesting to do that.
Yeah, yeah, right.
So regarding the ACE2, generally, it's assumed that respiratory viruses particularly affect or predominantly affect the epithelium. Now, ACE2 is, of course, mostly a feature of the endothelium.
Yes, that's right.
Do you actually have relevant ACE2 expression in the alveolar epithelium, or does the virus actually infect the endothelium as well?
Yeah.
Or how does it?
Yeah, I think there's data out for SARS on that, and it's expressed, ACE2 is expressed on the epithelium, but it is speculated that there might be also an additional receptor because ACE2 is more confined to the distal lung and not so much to the upper respiratory tract. So maybe for this new coronavirus, there might be another receptor that is also used, but this is still, this work is to be done, right?
Okay. I was wondering whether you had any data on the interaction with this specific immunities with specific antiviral GM-CSF?
Yes, in the influenza model we did that. So we thought that we found that GM-CSF were specifically activating CD103 positive dendritic cells. This is the mucosal version of the dendritic cell. And this results in better generation of antigen-specific T cells. So we looked with tetramers and so on. So it's also antigen-specific.
So this would also work in viral infection?
Yes.
At certain
yeah.
stages.
Yeah. Actually, I mean, the data is not yet blinded because we are still recruiting, but we have a lot of patients with influenza in the trial, so we may see that eventually, because I mean, we sample, with the BIL, we sample those cells, right, and we store them and so on. So.
Yes, please.
So every now and then there's a new immune cell in the lung discovered producing GM-CSF, now basophils, I think.
Yeah.
Shaping macrophage responses. So
right.
Do you think the basophils also influence your stem cells?
Yeah.
How do you see this? Every cell makes GM-CSF and specifically cares about them.
Right. We have flow sorted all the different subsets that occur during influenza infection, both the parenchymal cells, like with type 1, type 2 alveolar epithelial cells and so on, and all the immune cells. And we have seen that in influenza, this is only in the influenza model, the cell type that expresses it most highly is in fact type 2 alveolar epithelial cells. So if you put that side by side, you can see that. And there's not too many basal cells. So I think in that situation of severe pneumonia, the basal cell might play a minor role, right? So you refer to a paper that focuses on development of the lung and development of the macrophage pool in the lung in early or in late embryogenesis, actually, when the first breath occurs and the macrophage pool arises, and there is a crosstalk of basophils with GM-CSF, and GM-CSF is important for differentiation of macrophages, and it's said in that paper that this also prevents from pneumococcal pneumonia, or if you have more GM-CSF, you would be prevented from pneumococcal pneumonia in your very early days. But this is, I think it's another story because it's just a very different entity. Yes, yeah, exactly.
I have a question with respect to your experience with the funding of the clinical study, because what you said, it looks like this can happen quite often. So a company sold, investors don't think it's a interesting topic, and then in your case, the DZL, as far as I understood, stepped in. So. How about the structure we have to implement? Is it that we need kind of public funding to come in when PPP more or less fails, or what would be a rescue mechanism?
Right.
And because that seemed to be a little bit ad hoc, that DZL was there to help you.
Yeah, I mean, we have calls for clinical trials each and every year, so we were just happy to be in the timeline. But yes, of course, I mean, that was instrumental, otherwise we would not have been able to continue that. So you need kind of a funding source or a part of your funding provided for such scenarios. I think this is relevant. I mean, you cannot go for a phase 3 with that funding, of course. And of course, we have cross-funding by other sources because, I mean, it's quite an expensive trial. So ARDS trials or intensive care trials are really expensive. Like it's the case money is 10,000 euros. So that's quite some number. But yes, I think you need to have these kind of emergency funding sources. Yeah.
Okay. Yeah.
Just following on that, do you have any strategy in your system to de-risk by, for example, a health economic assessment or something that you have new arguments for the next potential investor? Because I guess you will not sell it on the market in the end. You need at some point an investor to come in.
Yes, you're right. Yes, you're right. I mean, the only strategy that we can go in that way is just the precision phenotyping and say, okay, what is new with us? Why do we think that our ARDS trial is going to be successful? And this is because we phenotype it. We don't see ARDS as all the other trials. And look, this is our strategy, and these infrastructure or structural infrastructure we have in place provides us with that information. I think this is something that would help.
Because it would be a perfect chance in your current trial to collect the data and
right.
at least bring internal budgeting data.
Yeah, exactly. Right.
Okay, then if there are no further questions, we are just in time, and thank you very much again.
Thank you.